Welcome to the Deep Dive Books channel, where we hope you find something valuable every day.
Let's continue with Max Bennett's A Brief History of Intelligence. This is the third part of our exploration. In the AI world today, there's a saying: the architecture of a model doesn't matter much. Any structure will suffice, as long as you have enough computational power, large datasets, and numerous parameters. The model can learn anything. However, when we examine the evolution of biological intelligence, it's clear this isn't the case. It's not just about how fast the brain can process information. Without the right structure, intelligence doesn't emerge. Often, in the history of evolution, a random fork in the road led to one branch missing out on higher intelligence entirely. Today, we'll discuss the second major breakthrough in intelligence in Max Bennett's narrative: vertebrates. Since the emergence of bilaterally symmetrical animals on Earth, the biological world entered into an arms race. Species developed various strategies for hunting and evading predators, leading to the so-called Cambrian explosion, a rapid diversification of species. Around 500 million years ago, there was a split in the evolution of nematodes, with one branch leading to vertebrates, and the other to invertebrates. In the invertebrate branch, arthropods emerged as the dominant force during the Cambrian period. Marine arthropods could grow to be 1 or 2 meters long, quite formidable creatures. Unfortunately, this branch, including all invertebrates, didn't have much potential for further development. Even today, the smartest arthropods are crabs, ants, and bees. Luckily, we belong to the other branch, vertebrates. The earliest vertebrates were small, fish-like creatures, only a few inches long, almost insignificant in the competitive Cambrian world. But the main structure of their brain was almost identical to that of modern fish and humans. A cortex, basal ganglia, thalamus, hypothalamus, midbrain, and hindbrain. The only difference between our brain and theirs is that we have evolved a neocortex, which was a later breakthrough. So, what new intelligence did this advanced brain bring to vertebrates? The answer is learning. Vertebrates were faced with a highly competitive world, a complex environment, where the reflex-based learning of nematodes was far from sufficient. They needed to learn more advanced skills. In the late 19th century, a young psychologist named Edward Thorndike, 1874 to 1949, wanted to understand how animals learn. He designed a series of cages, mazes, and other devices, using chicks, kittens, puppies, and even fish for experiments, to see how they could learn to escape and get food. In some cases, they needed to figure out the right path; in others, they had to unlock something, press a button, or perform a specific action. Like a kitten licking itself, before Thorndike would open the door for them. Thorndike initially thought animals would learn by imitation. Watching others, remembering, much like how we learn in class. But he discovered that this wasn't the case. They didn't learn just by watching. The animals would first explore on their own, moving here and there, occasionally succeeding, and getting a reward. Over time, they solved the same problem faster and faster. This is what we call trial and error. Even fish have this ability. Many people believe fish only have a 3-second memory, but that's not true. Fish can learn through trial and error how to navigate a complex aquarium maze and, even years later, remember the original path. If even fish can do this, AI should be able to as well. The first wave of AI enthusiasm, in the 1950s, led researchers to borrow from Thorndike's findings, designing neural networks that could learn to navigate mazes and play chess through trial and error. The idea was simple. Try whatever you want, and if you win, get a reward. If you lose, face a penalty. Let's see what you can learn. As you might expect, this is what we now call reinforcement learning. However, scientists soon discovered that this method only worked for simple problems. For slightly more complex tasks, like chess, where you need to make hundreds of moves to reach the end, it's challenging to learn from such a long sequence of steps. For the next 3 or 4 decades, no one could solve this problem. It seemed simple, but people didn't really understand how fish managed to learn through trial and error. It wasn't until 1984 that a brilliant mind, Richard S. Sutton, solved this problem. We've talked about Sutton on this channel before. He studied psychology as an undergraduate and found inspiration there. Sutton's insight was that you shouldn't use the final result as the sole reward, but rather consider the expectation of the result at every step as a reward. Simply put, Sutton broke reinforcement learning into two independent parts, each trained separately: the actor and the critic. After each step taken by the actor, the critic predicts the overall probability of success at that point. Sutton suggested that we should reinforce not just the final outcome, but the change in the probability of success after each step. For example, if the probability of winning was initially 51%, and after taking a step, the critic says, it's now 61%, then that step should be considered a good move, and reinforced. This way, the learning process isn't just about the final win or loss, but about evaluating each step as good or bad. Even if you lose the game in the end, you've still learned many good moves along the way. This is known as temporal difference learning, the fundamental principle of reinforcement learning today, including in systems like AlphaGo. In fact, as early as the 1990s, someone used Sutton's method to create an AI that could play backgammon at a very high level. So, do animals learn this way too? Yes, animals also use temporal difference learning for trial and error learning. This is one of the most elegant examples of how AI research has informed neuroscience, helping scientists truly understand dopamine. It turns out that dopamine is the key to reinforcement learning. Initially, researchers thought dopamine was a reward substance. In experiments, when a monkey was given some sugary water, its brain would immediately produce dopamine, suggesting that dopamine represented liking. But after repeating this a few times, they discovered that the monkey's brain no longer released dopamine after receiving the sugary water, but before, when it anticipated getting it. Dopamine is about the anticipation of good things, not the reward itself. This anticipation can be quantified. Closer and more likely the good thing is, the more dopamine is released. This is exactly how Sutton's temporal difference learning algorithm processes signals for reinforcing AI. In 1997, a paper combined AI principles to thoroughly explain how dopamine works. Dopamine is a reinforcement signal, not a reward signal. Its role is to make us want, telling us that something good is nearby, that what we're doing is correct, and encouraging us to keep going. We mentioned earlier that even the earliest bilaterally symmetrical animals, like nematodes, had dopamine. But their dopamine system is more primitive, only signaling that something good is nearby. In contrast, vertebrate dopamine is a quantifiable signal. The more dopamine, the higher the likelihood, and the closer the timing of a good event, boosting our motivation. An essential factor here is the sense of time. Vertebrates have a sense of time and can accurately perceive the duration between two events. Invertebrates, even more advanced ones, like crabs and bees, cannot perceive time intervals, significantly limiting their learning ability. In the vertebrate brain, the hypothalamus releases dopamine. It's a reward system that only looks at outcomes and releases dopamine if something is deemed good. However, the brain's actual learning mechanism isn't about releasing, but sensing dopamine, handled by the basal ganglia. The basal ganglia have two circuits, one acting as the actor, and the other as the critic. The critic senses dopamine, and together they learn. When a monkey first drinks sugary water, the hypothalamus releases dopamine, and the basal ganglia recognize that this event is worth learning. After a few repetitions, learning becomes more precise, with the basal ganglia circuits learning to judge the probability of a reward, thus quantifying dopamine perception and forming reinforcement learning. But the story doesn't end here. Reinforcement learning algorithms have achieved tremendous success, allowing AI to surpass human performance in many video games. However, there's one game, Montezuma's Revenge, released in 1984, where AI struggled to keep up. This is a maze-like game, where the player must navigate a room full of obstacles to find a hidden door. Unlike other games, the hidden door can be anywhere, and there are no milestone steps. You have no idea if you're doing something right or wrong until you find the door. This makes it very challenging for reinforcement learning. To try new things, reinforcement learning usually allows the actor some random actions, perhaps around 5%, but that seems insufficient here. Yet if you wander aimlessly, you'll find the door too slowly. It wasn't until 2018 that Google's DeepMind team finally conquered Montezuma's revenge, beating human players. Their solution was to introduce curiosity. We've discussed this concept on our channel before, when reviewing the book, Why Greatness Cannot Be Planned, The Myth of the Objective. It's called novelty search. The core idea is that, if an action doesn't bring you a reward, but is novel and explores an unknown area, satisfying curiosity, it should still be reinforced. AI, driven by curiosity, actively explored uncharted areas of the room, and eventually found the hidden door. Vertebrates, as well as some advanced invertebrates that evolved later, possess curiosity. We can receive dopamine just by satisfying our curiosity. This is why we are so easily attracted to random rewards, and why we can remain so engaged in a casino, even while losing money. Reinforcement learning has clear goals. It's a very utilitarian approach. However, it must be guided by curiosity to go far. You must be willing to sacrifice some rewards just to explore new possibilities. Curiosity makes the act of learning itself a worthwhile pursuit. It turns out that reinforcement learning and curiosity are the easiest forms of learning to understand. However, vertebrates have some particularly important learning abilities that we still don't fully comprehend. That is pattern recognition. Yesterday, you encountered a big fish in the deep sea and nearly got eaten. You remembered it. Today, you encounter another creature that looks similar. How do you know if it's a predator like the one you saw yesterday, or a potential mate? It's like when a human walks down the street and smells a mixture of proteins and carbohydrates. How do you know whether it's a steamed bun or something inedible? These are examples of pattern recognition. Vertebrates don't assign a specific neuron to each object. The information we receive is complex and overlapping, yet we can recognize patterns with precision. How do we do that? For AI, the solution is supervised learning. This is the masterpiece of Geoffrey Hinton, a prominent figure often in the spotlight, and the mentor of Ilya Sutskever, the former chief scientist of OpenAI. You create a large neural network, pre-label each training material, and update the network parameters backward, based on the output, after each training session. Supervised learning algorithms have been highly successful. For example, your phone can recognize your face. Even if your face looks slightly different today than it did yesterday, it knows it's still you. And if someone who looks very similar to you comes along, it knows it's not you. However, the vertebrate brain does not use supervised learning for pattern recognition. No one labels training materials for them, and a carbon-based brain can't adjust the parameters of numerous neurons with each training session. Vertebrates rely on automatic associative learning for pattern recognition, but we won't go into the details here. A famous problem in pattern recognition is illustrated like this. Two pictures, each containing two objects. A rectangular prism and a pyramid. At a glance, you can tell that these are likely the same objects viewed from different angles, right? Even fish can recognize this. But for computers, it's extremely difficult. The image looks very different when the angle changes, so how do you know if it's the same object from a different perspective, or two entirely different objects? AI scientists eventually solved this problem using convolutional networks. But we know for sure that human and fish brains do not use convolutional networks. Our neurons are not structured in so many layers. What method does the brain use? We don't know. Moreover, after being trained, all large language models, including the most advanced ones like GPT, must have their parameters locked. Before being released to the market, they cannot continue training freely. This is because the training must be done very carefully. If not, learning new knowledge could overwrite old knowledge, leading to catastrophic forgetting. However, vertebrates never experience catastrophic forgetting. A fish doesn't forget an old skill just because it learns a new one. We are masters of many trades. How do carbon-based brains avoid catastrophic forgetting? No one knows for sure. But we do know that, achieving certain specialized functions, requires specialized hardware structures. For example, the hippocampus in vertebrate brains is crucial for our ability to form spatial awareness. When you go out, you don't follow the same route every time. You take shortcuts, because you've built a map of the world in your hippocampus, allowing you to know where you are. Bees and ants lack this ability. The most significant insight from this breakthrough might be the relationship between reinforcement learning and curiosity. It turns out that trial and error is a more fundamental learning ability than imitation, and curiosity is not just an optional emotion. It's a key survival strategy. If that's the case, then some modern educational approaches, which confine children to classrooms where they listen to teachers, memorize a bunch of facts, rarely engage in hands-on activities, discourage trial and error, and even forbid reading extracurricular books, are stifling the innate nature of vertebrates, aren't they? I am a vertebrate, and I'm proud of it. That's all for today. In the next part, we'll discuss imagination.
If you feel there is value in this, please like, subscribe to this channel, and leave your thoughts or suggestions in the comments section. Let's grow together and read more good books.